Hi folks, and welcome to another edition of Zog Science. Today we're going to be introducing our biochemistry section of this unit, and we're going to be specifically talking about carbon and kind of the basics of the biochemistry. So, living things comprise mostly of carbon-based compounds, except for water, which obviously makes up the majority of living things. And carbon is very diverse and very unique in that it can create lots of diverse molecules. So that ability to create large and complex molecules is what makes carbon such an important compound. And compounds containing carbon are said to be organic compounds. And so the main four classes of organic compounds are carbohydrates, lipids, proteins, and nucleic acids. We'll talk about each of those more specifically in the next video, but this one we're just going to be talking about carbon itself. So carbon atoms can form lots of diverse molecules and by bonding to at least 4 other atoms. And the reason why it can do that is because of its valence electrons. Recall from chemistry that carbon has 4 valence electrons, and because of that, it can form up to 4 bonds, and it can form those bonds with a variety of atoms. It's not just it's not designated to make bonds with only certain kinds of atoms the way that other ones are. So the ones that it partners with the most are hydrogen, oxygen, and nitrogen. And along with carbon, those 4 molecules are some of the major components of organic molecules. We'll also get some sulfur and some phosphorus, but generally hydrogen, oxygen, and nitrogen and carbon are the main elements that make up living things. And part of the reason why carbon is such a key thing and why we can get so many varieties of organic molecules is that we can get such a wide variety of different types of bonds, the way that things bond. So we can get different length carbon chains. We can get double bonds in different positions. Remember, structure determines function. So having that double bond in a different place can determine a different function. We can also get branches, and then finally we can get different kinds of rings. So this variation is what makes carbon such a useful element for living things, because you can get a wide variety of molecules. Now, one of the things that we can have is we can also have compounds or molecules that have the same chemical formula, the same number of carbons, hydrogens, oxygens, but they are in different arrangements. They have a different structure, and so because they have a different structure, they're going to have a different function and different properties. There's a few different types of these isomers. We can have structural isomers. So, for example, rather than just having a long chain, maybe there's a branch. We could also have cis-trans isomers, which are basically situated around a double bond. And we can also have enantiomers, which are mirror images of each other. Now, it's not super important to me that you know all of these specific types of isomers. It's much more important to me that you just know overall that there are isomers. And the reason why it's important is that molecules, again, when they have different structures, they have different functions. So, for example, back in the 50s and 60s, there was a medication that was given to women for morning sickness, and one of the enantiomers of that actually did help with morning sickness. Unfortunately, the other enantiomer of that was causing birth defects, and so they had to discontinue the use of that drug, and they had to learn how to be able to separate out drugs by their enantiomers and make sure that they test both of the isomers for toxicity. And what we find is that in living things, most of the sugars are one type of isomer, and then amino acids are the other type of isomer. So pretty interesting. Now, when we look at carbon chains, right, carbon chains are just carbons and hydrogens, not very interesting, but functional groups are kind of chemical groups that we see reoccurring, and they have specific functions that help provide molecules with their different structures. So, for example, if you take a look at estradiol, which is a type of estrogen, and then you take a look at testosterone, they have the same general shape, right? You kind of see the different ring shapes. They have the same methyl group and hydroxyl group over here on this end, but testosterone has a slightly different set of functional groups here on this end of the molecule and this one, estrogen has a different set, right? That is going to help to determine their function. So it's important that we know these functional groups. These are the 6 kind of most important functional groups. The ones that I think are the most important, the ones that you should definitely make sure that you know, are the hydroxyl, carboxyl, amino, and phosphate groups. We will talk a little bit about the sulfahydryl and methyl groups, but we won't get into them nearly as much as we will the others. So to start off with hydroxyl groups, that's just an oxygen bound to a hydrogen. It's an alcohol, and usually a molecule that has this is going to have an OL, for example, ethanol, right? The reason why this hydroxide group is important is that it can form a lot of hydrogen bonds. So whenever you see that, you should think that A, the molecule might be polar, and B, that there's the possibility for hydrogen bonds. We've also got our carboxyl group, sometimes called the carboxylic acid or organic acids. The reason why is that hydrogen can be donated, and if you recall from chemistry, your definition of an acid is a proton donor. So that hydrogen can be donated, and that's what makes it an acidic group. We see this on amino acids, which is the reason why we want to talk about it a lot. We'll see it in other molecules as well. So make sure that you know the carboxyl group. Then we've got the amino group. Amino group is kind of the opposite. It is the nitrogen and 2 hydrogens, and it is a proton acceptor. It can accept a hydrogen, which makes it base. And here is an amino acid. You'll notice that on one side we've got the carboxyl group, and on the other side we've got the amino group. And so in its natural state, this hydrogen is actually donated over here to this nitrogen. And so overall it's a balanced molecule, but it does have charges on both sides. And again, this is something that can form hydrogen bonds, and again is going to be charged, so this would be an indicator of a molecule being polar. And then our next one is the sulfahydryl group. This is a sulfur with a hydrogen, and the reason why it's important is that in protein structure, the sulfahydryl groups will form covalent bonds with each other, which will help with protein folding, which we'll get into later in this unit. Then we've got our phosphate group. Phosphate groups are found in DNA, which is why it's super important. It's also found in ATP. So again, very important that we understand this. Phosphate groups are a phosphate and then oxygens bounded all the way around. It's very negatively charged, which will be very important when we get to DNA. And then finally, we've got our methyl group. Methyl group is just a simple carbon with 3 hydrogens. Methyl groups can be added to DNA as markers, and we will talk about that later when we discuss epigenetics a little bit. We just want to make sure we understand what a methyl group is. All right, that's all I've got for you this time. We'll see you next time.